Now, most people are now familiar with the Watson-Crick model of DNA, the so-called B-form DNA. But real DNA is much longer than this and is indeed flexible. So I can illustrate that with a model. Now, this model, in fact, I hope you can see, is actually garden wire, and it is completely circular. And what I'm going to do is break one of the bonds, one of the phosphodiester bonds in DNA. And what I'm now going to do, show you, is actually rotate the broken end around the intact strand. And if you see elsewhere in the molecule, the DNA has coiled about its axis, or supercoiled. And supercoiling is one of the very common properties of DNA and has to be controlled. And that's controlled by the DNA topoisomerases. And if I simply take this model and cut it again and actually twizzle the ends in the opposite direction, the supercoils have all gone. The DNA circle is now relaxed. And this is a very common reaction that's carried out by DNA topoisomerases. And it's shown on the whiteboard here, the interconversion between a relaxed DNA circle and a supercoiled DNA circle. What I showed you, this nicking of the DNA and free rotation, is in fact one of the mechanisms that's used by DNA topoisomerases, but it's not the most common one. The most common one is in fact a mechanism called Strand Passage, which I'm going to show you now using a shorter DNA model made out of garden wire. What I'm going to do is not just break one strand, but I'm actually going to break both strands. And I'm going to take another piece of DNA, you can see it's already supercoiled, and pass it through that double strand break. And when I've resealed that break, the supercoils have gone. So strand passage is in fact another mechanism, and the more common mechanism, that DNA topoisomerases adopt. Now, you'll note that both these mechanisms involve breaking the DNA backbone. This is of course very useful. It allows you to do a number of DNA transactions, such as removing or introducing supercoils, or taking knots out of DNA, or decatenating DNA, that is unlinking linked circles. But it's also very dangerous. You must reseal the broken phosphodiester bonds, otherwise there'll be a break in the DNA and that can lead to cell death. Now, this single or double strand break process has been exploited by a number of anti-cancer drugs and antibiotics, which are able to stabilize the single-stranded or double-stranded break in DNA, and turns out to be a very effective way of killing cells, either anti-cancer cells in one case or bacterial pathogens.
Hello, my name is Keir Newman. I'm a co-author with Shannon McKee and Tony Maxwell on a BioEssays article covering the activity and function of topoisomerases. One of the aspects that we covered in the article are single molecule approaches to measurement of DNA topoisomerases. And I thought I would give you a brief overview of one of those techniques that we refer to as magnetic tweezers. It's emerged as a very powerful tool to measure the activity of topoisomerases. And so sitting next to me is one of the magnetic tweezers we built in my lab. It doesn't really look like it, but in fact it's just this inverted microscope. So I have an illumination system that passes through a microscope flow cell, which is really just a microscope slide. The light after that is collected by a microscope projectile that's sent to a CMOS camera. And the important aspect of this is that there's a pair of permanent magnets that are in this little contraption right here. And those apply an upwards magnetic field on anything I put inside that little thin flow cell. And so to think about how that works, now I'm going to sort of zoom in. This is now a DNA molecule. I can attach a single DNA molecule to the bottom of the microscope flow cell. And on the other side, I have a 1 micron magnetic particle. And because of the external magnets, it's experiencing a force upwards. I can also spin those magnets. And when I spin those magnets, I can now introduce supercoils into the DNA molecule. And you'll notice as I supercoil this, that for this plectinine to form, my hands have to get closer and closer together. And so using the inverted microscope, I can measure the position or the height of this magnetic particle above the surface, and that gives me a handle on the DNA topology. And so now as an enzyme unwinds this, you'll see my hands separate and I can rewind it, my hands get closer together. And so by measuring this separation, I can now measure the topology in real time, and by controlling the magnets, I can control the topology. So a typical experiment, I wind this up, I see my bead gets near the surface, and now the topoisomerase unwinds this, and I can see the bead jump up. And I can keep doing this process over and over again. And so by doing this, we affectionately refer to this as the topological treadmill. And so a poor enzyme, as I wind this up and wind this up, the enzyme starts unwinding and I rewind, and the enzyme unwinds and I rewind, and then we continue in this process through hundreds or thousands sometimes of supercoils relaxed or removed by individual topoisomerase. And the other experiment we can do, one other experiment we can do, for example, now is I can just take a long piece of linear DNA in which I've introduced a short single-stranded region, and one topoisomerase, a type 1 topoisomerase, will bind. And as it cuts that single-stranded region and opens it up, held together now just by the protein, this will extend a little bit. And so the bead will move up and move down as that gate, that protein-mediated gate, opens and closes. And I can now measure this process in real time. And what's more, I can control the kinetics of that by changing how hard I'm pulling with my external magnets. And so I can control that and now measure very accurately the force-dependent kinetics of that process. So that's just a very brief overview of one of the very powerful single molecule approaches used to study DNA topology and DNA topoisomerases. It's one of many approaches, but I thought this would give you a little bit of insight into how this particular single molecule method works. Shown here is a schematic of the magnetic tweezers measurement technique. The video shows a magnetic bead attached to a DNA molecule, schematically represented by the cyan circle attached to the line representing the DNA. Rotating the blue and red magnets supercoils the DNA, introducing a plectinine and decreasing the height of the bead. The height, or extension, of the bead is measured in real time, decreasing as the magnets are rotated and increasing as the topoisomerase relaxes the supercoil.
Hi, my name is Shannon McKee. I'm one of the authors of the recent BioEssays reviewing the DNA topoisomerases, and one of the main take-home messages from this work is that recent research is suggesting that in vivo, the topos are often forming parts of these highly complex multi-protein assemblies, which allows them to have much more far-reaching roles in various DNA metabolic processes. An example of this is that until recently, the only topo we thought to be directly involved in the generation of positive supercoils is a protein called reverse gyrase. This protein typically arises in organisms that must adapt to incredibly high temperature environments. However, Topo 3 alpha has been demonstrated by Bizard et al. To work alongside a protein called pitch. So pitch is a DNA translocase, and the hypothesis from that work was that as pitch translocates along the DNA, it extrudes highly negatively supercoiled DNA, which is a prime substrate for Topo 3 alpha. So Topo 3 alpha will relax those supercoils, and then when both the two proteins separate from the DNA, what you get is an accumulation of positive supercoiling, and it's thought that this happens because it promotes efficient centromere resolution. Another example as well is that Topo 3 beta has been demonstrated to interact with many RNA binding proteins such as TDRD3 and FMRP. And the interaction of Topo 3 beta with these proteins is suggesting that actually in vivo, the role of Topo 3 beta may actually be as a topo, an RNA topo rather than a DNA topo, which has actually excited a lot of research in that particular field. So please read our paper if you want to know more about the DNA topos and what they're up to in the cell, because after 50 years of research, there's still some very exciting and new data coming out that just completely demonstrates how important the topos are in pretty much every aspect of DNA metabolism.